Hey, Bio30s, and welcome to the final chapter of the course, the final unit of the course. We're going to be discussing genetic diversity in populations, otherwise known as population genetics. The section of the textbook that you want to be referring to is going to be chapter 19 of the textbook, and that's going to be covered on pages 676 to 701 in the textbook. I want to start today with a topic entitled Describing Genetic Diversity in Populations. And I want to take a step back last year to the unit on natural selection and Charles Darwin. I really feel that this unit should have been kind of back to back with that unit because they do kind of play very well together. This is sort of the science behind the observations that Darwin made when he was doing his journey around the world, when he came up with his theory of natural selection, where organisms in a population that have various variations, if a certain variation is more favorable in an environment, the organisms that have that variation are more likely to reproduce, to pass that variation down to their offspring. We now know how that occurs. It's through the process of the transfer of DNA, through the formation of gametes. We've been spending pretty much half the course this year discussing that. So how do we marry these two concepts together? Because really when Darwin was doing his work, Mendel was doing his work, neither one of the scientists really knew about the other's work and didn't really realize the ramifications of what the other scientist was discovering, because really these two branches of science, genetics and natural selection, play very well together. Because it was discovered that when you married the two concepts together, you could essentially explain how natural selection was occurring genetically, that you could see changes in a population over time, and that could eventually lead to a new species if that change was great enough over a long enough period of time. You might remember in Bio 20, one of the big things I said about natural selection was individual organisms don't change. It's populations that change. And what population genetics is about is to basically quantify the change. How do we quantify how much of a change is occurring in a population? Well, we basically look at their DNA. We look at the genes in the population of a specific trait. So I'm going to use the example that I always use, hitchhiker's thumb. So let's say I want to know in a population how many people have hitchhiker's thumb. Well, I could count 100 people, and let's say 50 people had hitchhiker's thumb, 50 people don't. I know it's controlled by a single gene. I know that gene has two copies of the allele. There's a recessive and a dominant allele, one that's going to cause a regular thumb, right? This is the recessive trait. Hitchhiker's thumb is the dominant trait. So I would have two recessive alleles in my DNA, so I'm going to be homozygous recessive. Well, the people who have hitchhiker's thumb in the population, the other 50, some of them are going to be homozygous dominant. Some of them are going to be heterozygous because it's a dominant versus recessive trait. Can I calculate the change in a population over time? Like if I were to look at that population, there was 50% hitchhiker's thumb, and then I come back 10 years later, look at the same population of humans in the same area, and now the population has 75% hitchhiker's thumb and only 25% don't have hitchhiker's thumb. There's obviously a change going on in the genetics of that population. Now, that begs the question, I've said this word about a billion times already today, what's a population? This is going back to Bio 20 as well. Bio 20, you might remember, a population is a group of organisms of the same species living in the same area at the same time. So technically our class, you could count the people in our class in our room, that would be a population of humans living in, let's say, 111 or wherever we're at, right? That would be considered a population. If I counted that population, let's say there's 30 kids in the class, and 20 of them have hitchhiker's thumb, 10 of them don't. I can quantify the genetic makeup of that population for that particular trait. What I'm discussing there is called the gene pool. It's basically the sum of all the genes that are found in that population of organisms. So in the case of 30 people in the Bio 30 class, and there's 30 kids in there, and that's the population, technically there would be 60 copies of the gene, because each student would have two genes that control hitchhiker's thumb, right? You have them in pairs, right? So the total gene pool for hitchhiker's thumb in a class of 30 would be 60. There would be 60 genes in total. And could I then arrange and count those genes? Well, yeah, I can. And if I can do that, and if I can see if there's been changes in the structure or the percentage of the genes, how many are dominant, how many are recessive, then I can see if there's an actual change going on in the population. And that's what we're going to get to in this unit. It's a concept called Hardy-Weinberg principle, where we can actually, based on what the phenotypes of the population is, we can calculate the genotype ratios in the population, which I'll be getting to not today, but today will be sort of a setup for that. Yeah, so that's basically a population, and that's what a gene pool is here with our Mr. Deadpool there, our gene pool. So our gene pool would be, again, the sum of all the genes in a certain population. And how do you measure that? Well, there's three frequencies that you're going to use to measure a gene pool. And the three frequencies are what we call the genotype frequency. We've got the phenotype frequency, and we have the allele or the allelic frequency. So we've got the frequency of the different genotypes that would be found in a population for a specific trait, the phenotype frequencies, the different phenotypes that would be shown based on the genotypes of the organisms, and the actual frequency of the alleles. So how many copies of the recessive allele in total would there be in the population? How many copies of the dominant allele would be found in the total population? Let me give you an example. On your sheet, on your notes here, if you haven't got it up in front of you, maybe pull the notes up right now just to take a look. I've given you an example here of a population of mice. The mice, there are 200 individual mice in the population. We know just by looking at the genes that we have 72 of them that are homozygous black. Black is the dominant trait over white coloration. So the homozygous black ones that are 72 out of 200, they're going to be my homozygous dominants. I've got 92 that are going to be heterozygous. They're going to have one copy of the dominant and one copy of the recessive allele. So that's 96 out of 200 are going to be in that category. And then 32 out of the 200 are known to be white, and they have the genotype little b because it's the recessive trait. Just by taking the numbers of each of those groups of organisms and dividing it by the total population, I can calculate my genotype frequency. So in this case, my population, in the population, the homozygous dominant frequency would be 36%, or a frequency of 0.36. My heterozygotes would be at a frequency of 48%, or 0.48 as a frequency, and my homozygous recessives, my little b's, they're going to have a frequency of 16%, or 0.16. So that calculates all my genotype frequencies. That's actually tougher to calculate because normally, if you get a population of organisms, you can't tell whether they're homozygous dominant or recessive, or heterozygous, just by looking at them. Normally, what you can calculate right off the get-go is the phenotype ratio. The phenotype ratio would be the different variations that you would see of that particular trait. In this case, white or black fur. Well, I know that in this case, I was given my genotype frequencies already, which makes it easy. My two genotypes, my homozygous dominant and my heterozygous configurations, they're both combined going to give me organisms that have black coloration. So I have to take 36 plus 48, and I'm going to get a frequency of 84%. So my black phenotype, which represents the homozygous dominant, the heterozygous configurations of the genotypes, that frequency of black in the population is going to be 84. All right? My white phenotype ratio is going to be the same as my homozygous recessive frequency up here of 16%, because only the little b's can be white. So in this case, I'm only going to have two frequencies present because there's only two phenotypes present for that particular gene. If I was doing, let's say, blood typing, that would be a little different. Then I would have 4 different phenotypes would be represented by the 4 different blood types. But in this case, and for this course, and for this unit, we're not going to be dealing with more than 2 possible phenotypes for any one of the questions you're going to get. So don't panic that you're going to get a bunch of questions that are going to have multiple phenotypes. That isn't going to happen, especially when it comes to calculating equations using the Hardy-Weinberg principle, which we'll get to in my next video. The final frequency is the allelic frequency, and this one can be a little bit confusing, but it's not really. There are 200 mice in the population. We know that each mouse has two genes that represent the coloration. So they each have two genes. I've got 1, 2, 1, 2, 1, 2, right? So if I've got 200 individuals for one gene or for one trait that's controlled by a single gene, technically there are going to be 400 alleles in the population because each individual mouse has two alleles for this single gene controlled trait. So in this case, I have to divide my numbers by 400. But how do I get my number of alleles? Well, that's kind of obvious, but I'll walk you through it. Capital B, those are going to be, my two alleles are simple. Let me start off with the number, let me start off with the alleles. The two alleles that are present are going to be capital B and little b. There's only going to be the dominant allele or the recessive allele. So I have to calculate how many times I see the dominant allele in my population. Well, I know my genotype frequency, so that's going to help me. I've got 72 individuals that are capital B. So that means I'm going to have 72 plus 72 because I've got 72 capital B's, and then I also have another 72 capital B's in those organisms. So I'm going to have 144 capital B alleles just present in the homozygous dominant organisms, 72 plus 72. But I also have to factor in the fact that there are 96 heterozygotes in the population as well. That means that I've got one set of 96 capital B's that I have to add. So I've got 72 plus 96 alleles out of 400 is going to give me an allelic frequency for the dominant allele of 0.60 or 60%. All right, that's how I calculate that. How do I calculate my recessive allele? Well, I could just take 0.60 and subtract it from 1.0 to get an answer of 0.4, but I should probably check, because if I do my math for the top and the bottom, remember, all these frequencies need to equal what number, right? 36 plus 48 plus 16 gives me a total percentage of what? Hello. It's got to be 100. If I get a number other than 100, I've done something wrong. All my frequencies have to equal 1.00 or 1.0 or 1. That is key. You can't have more than 100% of a frequency, right? The maximum you can have is 100%. So that number cannot be above or below 1. When you add up all the frequencies in the population, whether they're genotype frequencies, phenotype frequencies, allelic frequencies, those should always add up to a total number of 1 or 1.0 or 1.00. You can't have any other number. Otherwise, you may have done something wrong with the math. Let's see if our math works, though, here. I said the answer was supposed to be 0.4 because I took 1 minus 0.6. That's sort of a cheating way to do it, but I want to double check to make sure that I haven't done anything wrong. Little b, well, I've got 2 copies of the little b here with my homozygous recessives, so that's going to be 32 plus 32, but I also have to calculate that 96 organisms are heterozygous, so 96 organisms are going to have a single copy of the recessive allele. That's going to be 96 plus 32 divided by 400. Guess what? It's going to equal 0.40, hello, 40%. So I got 40% and 60%. That equals out my allelic frequencies, as you can see right there on the slide. Hopefully you're following along at home. If you don't like math, this is not your unit. There's going to be a lot of math in this unit, a lot more than you're probably used to in a bio class. So I'm just warning you in advance right now, this is going to be the most math you've had to deal with in any unit in bio up to this point. So just keep that in mind as we move into this unit. Here's a recap of everything I just described, showing you the phenotypes based with the genotypes and the allelic frequencies. They're all there. Again, I use those numbers here on the board as well, so hopefully that all gelled with the examples that were in the notes for you. Before we move on, let's compare geno versus phenotypes versus allelic frequencies one more time. Here you go. You've got a whole leaf here full of little bugs. Most of them are green. In this case, they're saying that 75 out of 100 bugs on the leaf are green. That's going to be the 0.75 frequency for green phenotype. 0.25 would be the frequency for the red phenotype. So my frequencies for my phenotype frequencies are going to be 0.75 for green and 0.25 for red, according to the bar graph right over here. They're also indicating that the genotype frequency breakdown, although we wouldn't be able to do it based on just the phenotype, at least not yet, You will be after today's lesson. I'll be able to show you how to do the math for it. But in this case, the breakup is 0.25 for homozygous dominant, 50% or 0.5 for heterozygous, and 0.25 for homozygous recessive. And then the allelic frequencies based on that information is going to be a 50-50 split. We're looking at a population that has half the dominant allele and half the recessive allele. So again, I just wanted to introduce you to the concepts of genotype frequencies, allelic frequencies, and phenotype frequencies, because this is all going to play a role as we move into learning how to use the Hardy-Weinberg equation, or wrapping our head around the Hardy-Weinberg principle, which I'm going to get to next day. And again, I'm kind of doing the segue to it right now, even though I'm going to stop the video here in a couple seconds. But yeah, Hardy and Weinberg were two guys that were involved in basically mathematically being able to sort of predict, if I knew the phenotype frequencies in a population, could I essentially calculate the genotype frequencies based on what I see with the phenotype frequencies? And the answer is yes. They came up with a very famous equation, a very simple equation, which is P plus Q equals 1. And that's basically what I'm going to get to next day when I discuss this little nugget, the Hardy-Weinberg principle. Until then, thanks for tuning in, and we will see you very soon.